Yeah. So, good afternoon, everyone. So, myself, Kundan Solanki. Okay. I'm one of your TA uh, in your cell biology course. Okay. And today is our second session, tutorial session. Okay. So, as I told in the first session as well, the idea of this session is to clear the doubts. Okay. For the from the content of the week which you had gone through. So, I hope everyone had gone through the content. Can I get like hands up? I mean, how many of you have done the content, gone through the content of the week? Yeah, okay, yeah, you can raise your hands or you can like on your audio so that it will be better for me to judge. Like if you have gone through the content, then we can revise it accordingly. Okay, so some of you have not gone through the content. What about others? Okay. Yeah, so basically we will be seeing uh, we will first go through the week's content, okay, and then uh, we will see some assignment questions. So, have you attempted the assignments question? Have you attempted the assignment, or you are you left with the assignment? You can on your microphone and can also speak. Okay. So,
Assignment is left, sir.
okay, assignment is left. So, what should we do? Can we discuss the assignment over here, or because what as I told in the earlier class also, if you attempt the assignment, otherwise what I can do is that I can bring you other sample questions and assignment question like you can do on your own. And because what I taught was that if you go through the week content before this class and you attempt the assignment, then we can discuss that assignment question and you can have doubt. So every week content and assignment we can discuss in that week only at the end of the week. That means on today. So anyways, like you can go through the assignment, okay, and we will go through some questions. We will go through some lectures first, okay, lectures content first, and then afterwards we can see the assignments, okay, other assignments question. Yeah, so basically this is second session, and so can anyone tell me the people which have seen the recordings, okay? So the class. So, can anyone tell me what you studied in that class? In the week two content, what you studied? You can turn on your microphone and can also speak. Okay. So, so in second week, generally we will be discussing like the cell cycle regulation and all those things. Okay. So if you have not gone through the lecture, we will just revise it over here. Okay. I mean, I will go through the lectures content
Sir,
content
mitosis, meiosis, G1, S1, all cell cycle. It was like I have attended half of them.
mhm, yeah. So the in the week two, basically, you dealt with the mitosis and meiosis process. Okay, so basically the experimentation. Okay, the CDK and cyclins, which are responsible for the cell cycle control and regulation in the mitosis and meiosis. Mitosis basically, not meiosis. So we will be dealing with the mitosis only currently. Okay, so meiosis will be dealing later on. But in this class, in general, in mitosis, what are the cell cycle regulations which are happening during the mitosis phase? Okay, so first we'll be talking about different phases in the mitosis. So when we talk about phases, there are like G1, AS, G2, M. Okay, so we will be looking about at that. And there are cell cycle regulations and various proteins involved over there. Okay, in that case, so we will be seeing what are the different proteins which are involved. So CDK and cyclin specifically, that are the proteins which are involved in the regulation of that mitosis and mitosis process. Okay, so we'll be looking at that, and yeah, and after that, uh, there was another uh, another lecture on the proteasome mechanism. Okay, so ubiquitin proteasome system. So how ubiquitin, what are the enzymes, structural configuration structures, okay, the enzyme, the ubiquitin, and what are the structure is made up of, and what are the residues which are involved over there, and how it functions in the regulation of cell cycle. Okay, so ubiquitination process and the regulation of cell cycle are very much interlinked, okay, to each other. Because as you know, ubiquitin mediates the degradation of protein, okay, basically by tagging the ubiquitin onto the protein molecule. So it is also a regulated process. It is not that randomly any of the protein will get ubiquitinated. It is a regulated process, and it happens during the certain conditions only when there are adverse effect or adverse conditions observed during the replication process or during the division process. Then only the such processes or such regulations occur. Okay, in the cell. So if you have not gone through the slide, if you have not gone through the content. So I highly suggest कि you go through the content first. Okay. In this lecture, basically the idea of this lecture is to not to discuss in detail regarding the whole content, which is provided in the week or in that specific week. The idea of this class is to discuss the doubt. Okay. So doubt will only arise after you have gone through the content of that week. So I would highly recommend today as well as in coming lectures as well that first you go through the content before this class, okay, and you try to solve the assignment question at least one or two tries, okay, and so that you will be aware of the content as well, and you will be after solving the questions, you will be having some specific doubt as well, okay. So after that, you will be better. This class will be very fruitful if you have any doubt, and we can try to solve it because if you if you come in the next class and ask that out of this class, then it will be very difficult for me as well to just go through back in the back class and then look what is the specific thing which you are asking. Okay, so as I told in the last lecture as well, and I am telling currently that you can go through the week content before Saturday, so that on Saturday we can discuss, go through overview. We can quickly revise the content and we can solve the assignment questions. Okay, so please maintain this thing from the next class. I would suggest that. Yeah. So first, as I told, we will be discussing about the cell cycle regulation. Okay, and various proteins which are involved over there and how it was discovered basically during the initial phase. So uh, the first stage, as we already know, what are the stages involved in the cell cycle? Okay, during the mitosis phase, there is G1 phase. Okay, that's called gap one phase. There is an ACE phase, that is a DNA synthesis phase, where the replication occurs of the DNA by the sister chromate by the DNA molecule. Okay, and then there are G2 phase. Okay, which is a gap phase in between the S phase and the M phase. Okay, and after the G2 phase, there is a mitosis phase. Okay, so in the mitosis, basically the DNA which had replicated in this DNA in the S phase, it will divide it into two daughter cells. Okay, so mitosis will help in division of these two daughter molecules from a single parent molecule. Okay, so these are in general the cell cycle, and there is also a G0 phase that is called the non-cycling phase or the resting phase. Okay, in this phase actually the cell remain dormant. It will not undergo any change in the protein levels or any replication induced mechanism. So the cell will remain stagnant in that phase. Okay, so when there are limiting conditions in the environment or where there are adverse environmental factors which stops the reproduction of any organism, at that phase the cell enters into the Z0 phase and it inhibits the replication of the cells. Okay, so that it can maintain that survival in that condition. Okay, so basically, if I talk about the four different phases, so broadly we can classify that phase G0S and G1S and G2 phase as an interphase. Okay, so interphase consists of G0, G1, S, and G2. Okay, and after the interphase, there is a mitosis phase, and in mitosis, there are different phases like interphase, prophase, metaphase, anaphase, and telophase. Okay, so in interphase, basically the cells require cells. The in the interphase, basically the nuclear membrane, and uh, all those proteins which are involved in the maintaining of the nucleus will get disrupted. Okay, so basically the idea of mitosis is to segregate the DNA strand which is replicated into two daughter strand. Okay, two daughter cells. So in the interphase, the nuclear membrane and all those will be degraded, and in the prophase, the chromatid will be start to align with each other. Okay, so spindle fiber assembly will get started, and the spindle fiber and kinetochore will get formed. So kinetochore is an attachment of the chromatid. Okay, so here you can see, if you can see over here. Okay, here you can see the chromatids which are attached in the single plane. Okay, so these chromatids are attached by the kinetochore. So kinetochore is present in the on the chromatid through which the spindle fiber is attached. Okay, so spindle fiber will form, then there two polarities will be induced over here. Okay, left and the right polarity will get induced. In the metaphase, the chromatids will lie in the single plane. Okay, in the vertical direction, chromatid will be aligned to each other. And in the anaphase, basically the spindle fiber will pull the chromatid towards each other. Okay, and that is how the replicated DNA molecule, which is replicated in the synthesis phase, are segregated into two halves. Okay, at the left and the right half. In the anaphase, the spindle fiber contraction occurs. Okay, and formation of in the telophase, the cytokinesis occur. Basically, the cytokinesis is the separation of these two cells in single cell into two cells. Okay, and the nuclear membrane and the membrane will get developed, and two chromatids will get separated by each other. Okay, so yeah. So these are the things occurring in the mitosis phase, and in that case, the two chromatids will get separated to each other and forms the individual DNA molecule, individual daughter cells. And after that, again, it can enter to the interphase stage and again to the mitosis phase. So that is how the cell cycle replication occur, cell cycle division occurs in that case. Okay. And over here, you can see, as I talked over here, that there are certain regulations present in the cell cycle as well. Okay, so first is like G1 to S phase. There is certain restriction point because during the transformation of this G1 to S phase, there should be several certain proteins which should be present. Okay, after that only the cells will able to move into the S phase. If the protein is lacking, then the cells will not be moving into the synthesis phase. Okay, during the DNA synthesis. So when I talk about smoke, what are the certain factors which should be present? For example, in the S phase, the DNA replications are needed. Okay, so during that phase, a certain replication proteins should be present in the cell, or DNTPs and all those things should be present. Okay, and enzymes which are necessary for the replication of the DNA. That should be in sufficient amount during the synthesis phase. And if it is not that, there are certain checkpoints or regulatory proteins present that will not allow the transform of this G1 into the S phase. So that we will be studying that what are that regulatory proteins, okay, which limits the movement, which plays a role in the transfer of these cells from G1 to S and as well as S to G2. So these are called checkpoints, okay, between the cell cycle. So checkpoint proteins helps in maintaining that stability. So during the class, if my internet goes off, I mean, if I get disconnected, then please let me know, okay? Because I will not be able to know in between, and so please let me know if there is any fluctuation. Yeah, yeah. So the first thing what scientists studied in the regulation part was that he So they knew that there are certain cell cycle elements which are involved. Okay, synthesis phase, G1 phase, S phase, and G2 phase. So they wanted to check what are the what occurs if the both the two nucleus are present inside the same cell, and they wanted to check that what stage regulates at which stage. Okay, so they wanted to check that whether G1 is regulated by S or S is regulated by S regulates the G2 phase or whether G1 and G2 are interrelated to each other. Okay, so they wanted to check. And what they did that they took the two nucleus and incubated and transferred it into a single cell. Okay, and when they did that, and they found that the G1 nucleus was converted into the S phase. Okay, got translocated into the S phase. So they concluded that there are certain factors in the synthesis phase, S phase, which help the transformation of this S phase nucleus, G1 phase nucleus into the S phase. With certain proteins and other factors which will be present in this S phase nucleus helped in the movement of this G1 nucleus into the S phase. Another experiment what they did is they took the S nucleus and as well as G2 nucleus, and when they incubated in the single cell, they found that the G2 nucleus, the S nucleus continues to replicate on its own, but it didn't allow the transformation of this G2 nucleus. Okay. So from this study, what you can prove is that the proteins which are present in the S phase, okay, did not allow the movement of this G2. Okay, so G2 phase is not regulated by S phase. You can prove that thing from here. Okay. So there are certain other proteins which are required for transforming the G2 into the M phase. Okay. So these are the things. So from this also you can also deduce the regulation of certain phase and on which phase comes after which. Okay. So if the S phase nucleus is transforming the G1 phase, then we can say that G1 phase comes ahead of the S phase because the proteins which are needed in S phase are helpful in the G1 phase transforming to S phase. Okay, so this you can prove over here. And then when you to take the G1 and G2 both the nucleus and when you fuse it inside this when you incubate inside the single cell, so both the nucleus stays in that stage only. Okay, the G1 nucleus will enter S phase as per its own time, and G2 nucleus will remain as G2. Okay, so G1 will not help. G1 nucleus will not help the transforming the G2 nucleus into the M phase. Okay, so that protein is not present in the G1 stage. Okay, so here you can say that S phase is G1 phase is dependent on S phase. Okay, and G1 and G2 are not dependent on each other, and S and G2 also are S phase nucleus can also does not help in the G2 movement to the M phase. Okay, so here you can see that there would be certain replication promoting factors in the S phase. Okay, so that is why the in the G1 phase, in the G1 nucleus, when it got translocated into the when it was incubated with the S phase, the replication due to replication promoting factor in the S phase, it induced the replication in the G1 nucleus. Okay, and it induced the promotion into the S phase. Whereas S phase cell cannot induce the DNA synthesis in G2. Okay, because in G2 the cells are already replicated and there is no more needed. Okay, the DNA replication. So re-replication will not occur in the G2 phase, and as a result, the G2 phase nucleus will remain as it is. Okay, so this is the conclusion from this thing. Now, then another example of the regulation was found by this identification of maturation promoting factor. Okay, so maturation promoting factor is present which helps in the mitosis process. Okay, so when the cytoplasm from the M phase cell was incubated in the oocytes. So oocytes are basically a dormant egg, okay, which does not, which is present in the G0 phase, okay, in the resting phase. So when you incubate this G0 phase oocyte with the M cytoplasm of the M phase, in the cytoplasm there would be certain proteins factors present, proteins and other component which would be present in the cytoplasm of the M phase. And when you inject into the oocyte, that protein can induce the transformation of this oocyte resting phase into the mito M phase. Okay, so G0 phase will be converted into M phase. So here you can see that if you don't have certain proteins, then the cells will not get be dividing. Okay, and there will be certain proteins present in the M phase, which is helping in the transformation of this oocyte into the mature mitosis M phase. Okay, M phase transformation. So because that protein is promoting the mitosis, that is also called that is called maturation promoting factor. Okay, a mitosis promoting factor you can also tell. Okay, so that now you enter as a control experiment whether the cytoplasm from the M phase is inducing the division, or whether the interface cytoplasm can also induce the resting oocyte into the mitosis stage. They also incubated the cytoplasm from the interface stage. The interface stage, as I told, it contains G1 as and G2 phase. Okay, without M phase. So when incubated that cytoplasm, the oocyte remain in the G2 phase, but it did not transfer into the M phase. Okay, for M phase, there would be requirement of other proteins. Then only the oocyte will be moving from the G2 phase to M phase. So here you can say that there are certain regulatory proteins or certain regulations which is occurring, okay, which is not allowing the movement of the G2 phase nucleus oocyte into the M phase directly. Okay, and when that protein will be present, then only that G2 phase nucleus will be moving into the M phase. Okay, so that protein was called the maturation promoting factor, okay, which controls the mitosis stage. And these are the important protein which are if when it is present, then only the cell will undergo mitosis. And if you remove that, then the oocyte will remain in the G2 phase only. Okay. So here you can see various CDC mutants. Okay, so CDC, these mutants were identified in the Saccharomyces cerevisiae, or you can say budding yeast. Okay, so in the budding yeast first they were identified. So people from this experiment, basically the people tried got to know that there are certain regulations happening. Okay, and they said there should be certain regulatory proteins which would be involved in this from the conversion of this S phase to G2 phase and to M phase. Okay, so they found certain CDC mutant. They identified certain mutants in the Saccharomyces cerevisiae. Here you can see CDC1, CDC28, CDC14, CDC15. CDC1 and 28 basically in that mutant uh, the cells were not getting divided. Okay, So it was the DNA was not getting divided and it was not entering into the mitosis phase. The cells will not the cells DNA replication was not observed in the CDC28 and CDC1 mutant. Okay, so cells were getting arrested in the S phase only. Okay, in the DNA synthesis only. In the CDC14 here, you can see that the DNA replication was there, and the DNA was also segregated, but the cytokinesis step was not observed. Okay, so where you can see that the complete mitosis of the DNA of the daughter cells is not done. Okay, the spindle fiber is not getting got detached. Okay, CDC14 might be regulating that process, and if you mutate that CDC14, you can have the this kind of phenotypic observation. Okay, as well as the CDC15 as well. Here you can see that in certain cells, the DNA, the spindle fibers get disrupted. Okay, and the daughter cells have taken up this individual DNA molecule, but the but the separation of this separation of this yeast has not been done. Okay, which is present in the telophase basically. Okay, so in this you can say that CDC15 might be playing a role at this stage. So if I talk about this mutant as well, so these mutants are uh, basically simple to make. Okay, and this mutant is basically when you increase the temperature, this mutant becomes ineffective. Okay, so at the optimum temperature, these proteins will be working fine. Okay, and the DNA replication and the mitosis and all those things would be regulated properly. But when you increase the temperature, there will be this kind mutation in this. You can observe these mutants. Okay, or from there, and you can also observe this phenotype. So you can easily study that which proteins are getting affected. Okay, at the single stage. So like as I told, CDC28 is an important protein which is present during the cell cycle regulation. Okay, and so it is involved in the major cell cycle control at the start of the replication stage. So during the conversion from the G1 to S phase. Okay, so G1 to S phase, certain proteins are required. So at that stage, CDC28 works during the G1 to S phase, so that if it is mutant, then the cell will get uh, cell will not initiate the DNA synthesis basically. Okay, so CDC28 helps in the initiation of the DNA synthesis in the S phase, and in the mutant cell basically the DNA synthesis will not occur, and the cell will get arrested in the G1 phase only. Okay, so CDC28 basically codes for a cyclin dependent kinase that is called CDK. Okay, so CDK is a cyclin dependent kinase, and CDC28 is codes for that CDK. Okay, present in the yeast, Saccharomyces cerevisiae, and it limits the synthesis of the DNA and arrests the cells in the G1 stage. So then people also studied the. So is it clear to everyone up till here? I mean, is there any doubt? Hello.
Yes, sir.
Yeah. So uh, in between, like you need to respond. Okay. If you have any doubt, then you can directly ask. Okay. Yeah. So in the same way, they also studied CDC mutant in the Schizosacchomyces pombe. Okay, so this S pombe is a fission yeast. Okay, so basically in the cell cycle division, how it occurs. Here you can see the first the cell gets elongated. Okay, and that elongated cell will be there will be a formation of septum between the cell during the mitosis stage. Okay, during the division phase, and the cell will get divided into individual component. Okay, after that, so this is observed in the wild type cell, and in the mutation. So if you these are the three different CDC which was studied. Okay, CDC2, CDC25, and CDC13 mutant. Okay, so when you mutate these three CDC, here you can see that the cells are getting elongated, but there is no formation of septum over here. Okay, so cell division is not occurring, but the synthesis stage is occurring. Okay, so basically what you can infer is that this CDC plays a role during the mitosis stage. So DNA synthesis is not getting hampered, but the mitosis stage is getting hampered over here. Okay. So here you can get an inference that this CDC might play a role during the mitosis phase of the cell cycle. Okay, and another example here you can also see that CDC2D and V1. So V1 is an important protein over here. Okay, so V1 is an inhibitory kinase. Okay, we will be seeing further that how it inhibits the cell cycle regulation. Okay, so V1 basically inhibits the during the S phase. Okay, it inhibits the formation of a complete DNA synthesis molecule. Okay, so irregular septum will be get observed over here. Even if the cells is not elongated, as observed in the wild type, but there you will be forming the septum will be formed. Okay, so that regulation is hampered. Okay, over there. So in this V1 mutated S pombe. So So basically CDC2 encodes for a 34 kilodalton phosphoprotein. Okay, so this CDC2 encodes for a phosphoprotein that is P34, and P34 protein levels and phosphorylation do not change during the mitosis, but it becomes a dephosphorylated on entry to the stationary phase. Okay, so when P34 will be phosphorylated, then only the cell will get into the mitosis stage. Okay, and because CDC2 codes for a protein called P34, because we have a mutant of CDC2, P34 will not be present over there. And if the P34 will not be present, then the cell will not enter into mitosis. Okay, so that is how you can observe that regulation of CDC2 over here. Okay, and in the stationary phase, basically the P34 will remain dephosphorylated, and when you when transfer that into the mitosis phase. Okay, in the mitosis phase, it will get phosphorylated, and you will observe the mitosis, normal mitosis. Okay, and because so V1, as I told, uh, V1 is a phosphate. Okay, it induces the phosphorylation of P34. Okay, so sorry, it induces the dephosphorylation of P34 basically. So P34, when it is dephosphorylated, it remains in the stationary phase and cell can divide. But if it is phosphorylated, then cell undergoes the mitosis phase. So because P34 remains phosphorylated in the V1 condition, mutant condition. Okay. So V1 mutant condition, because it is a phosphatase, then it will not be able to remove the phosphate group from the P34, and P34 will remain always active. Okay, in that case. So that is why you can see that even though the cells are not getting properly divided, properly synthesis has not occurred, okay, DNA replication, even though the cell has undergone the mitosis, because the P34 is constitutively phosphorylated in this case, because the V1 phosphatase could not act on the P34. So this is the one regulation which which can be observed from this slide is that V1 regulates the P34 and induces the dephosphorylation. And dephosphorylated P34 then can lead to the DNA synthesis. Okay, and if the P34 remain phosphorylated, then the the cell will undergo mitosis stage. Okay, irregulated mitosis. So in the other study, people also studied in the sea urchins, the team hunt, okay, which was also awarded the Nobel Prize in that case. So he studied the maturation promoting factor in the sea urchins. So what he observed was that certain levels of cyclins, okay, so cyclins, certain level of cyclins was the cyclin is an activator, okay, it is a regulator of the CDKs, which I talked over here, CDKs, okay, CDC225 molar CDKs. So cyclin is a regulator for that. So when there is a they observe that there are certain proteins which was getting accumulated in the interface. It was increasing in the interface stage, and in the mitosis it was peaking, and at the end of the mitosis it was again coming down. Okay, so this was happening at every cycle. So in the interface it was getting synthesized. In the mitosis it was present in the peak level, and it was getting degraded. It was getting degraded at the end of the mitosis. Okay. So because he then observed that this protein basically helps in the transfer of this in helps in the cell cycle regulation. Okay, and it is involved in the mitosis phase of the cell cycle. Okay, so that he called mitosis maturation promoting factor. Okay, which helps in the maturation of this DNA, their cells. Okay, into the two daughter cells. So he termed this as maturation promoting factor. And now the maturation or the mitosis promoting factor, basically it helps in the inducing the immature oocyte to become an egg. Okay. So as I discussed in the earlier slide as well, that if you are incubating the cytoplasm from the M phase nucleus, okay. So as I discussed over here, that if you incubate the cytoplasm from the M phase cell into the normal oocyte, okay, dormant oocyte, that dormant oocyte will be converted into the mitosis phase. Okay, it will be transferred into the M phase, and cell division will be occurred. So there would be certain factors which are present in this M phase. Okay, and over here we are talking about that factor. Okay, what are these factors? Okay, so biochemical purification revealed that it has two subunits. It consists of cyclin dependent protein kinase, or also called CDK, and CDK and cyclin B. Okay, so CDK1 in case of humans, P34, CDC2. Okay, so that I discussed over here, CDC2, which encodes for P34 and which helps in the promotion of this mitosis. Okay, so CDC2, which encodes for P34 in Saccharomyces pombe, and CDC28 in the Saccharomyces cerevisiae. Okay, so the protein name are different, but it encodes for the it has the same role. Okay, and even the DNA sequence of this protein of these genes are complement to each other. Okay, so that experiment we will also be discussing, that when you have the DNA sequence of the CDC2, for example, and you take the whole genome sequence and you want to identify, people identify basically that whether there is any mammalian gene which corresponds to this DNA sequence, and they found that CDK1 was exactly complementing, binding with the CDC2 gene. Okay, so it was exactly complementing. So even the DNA sequence is similar, just that the namings are different over here. Okay, so don't get confused with these things. Okay, and so cycling dependent CDKs and cycling, it constitute the two subunits, and it does not work independently. Okay, so only CDK1, only if the CDK would be present, then it will not be functional. Okay, for its function, cyclins are required for it. Okay, and over here we are talking about cyclin B. Cyclin B are basically involved in the mitosis phase. But if I talk about S phase and G1 phase, there are different cyclins which are present. Okay, so that we will be discussing further in the further slide. But cyclin and CDK are dependent on each other. Okay, And even though the CDK during the whole process, the CDK will remain present, okay, in the whole stage. CDK will not get degraded, but cyclin level fluctuate, okay. Okay, the cyclin will get degraded and again reconstituted. So here you can observe the levels of these proteins. So he identified, Tim Hunt observed that there were certain proteins which was getting activated, increased, and it was peaking at the mitosis stage, and again it was getting degraded. So that protein was actually the cyclin. Okay. If we had observed the CDK, then CDK will not get degraded and again synthesized. Okay. It will be remaining as a flat line in this graph in the time dependent manner. Okay. So this you observe the cyclins in the basically in the sea urchins. Okay, and as I told, the cyclin is a regulatory subunit and it activates the CDK. The CDK on its own it does not get activated, but when it is bounded with the cyclin, okay, then only the CDK will get activated, and that will trigger the activation of CDK cyclin complex, and it will respond to cells. Okay, that there are certain proteins. So the cyclins and CDKs are regulated by certain proteins as well. Okay, so if there is any defects in the system, for example, we will talk in the further as well. If there is, for example, if there is a error in the DNA replication. Okay, so during the DNA replication, if there is a breakage or single strand breakage or double strand breakage, it will activate certain proteins which will inhibit the cyclin. Okay, it will degrade the cyclin, and it keeps the CDK in an inactive stage. Okay, so if the cyclin CDK complex is inactive, then the cell will get arrested in the S phase. Okay, in the synthesis phase, so it will not move ahead, because if you move ahead at that stage, then and if the cell is got by mitosis at that stage, then it will be a high chance that the DNA will not be getting separated equally at that stage, okay, because there is a breakage in the replication. So these are the regulatory molecules, cyclins and CDKs, which responds to the cellular condition, and various protein interacts with the various protein to observe that to say that the cells are not the replication system is not perfect currently, so that you will not be getting activated, okay, at this stage. But if the conditions are met, if the replications and all are happening fine, and there are various proteins and all are present in sufficient quantity for the mitosis stage, then the cyclin and CDK will get activated, and it will trigger the movement of the change of the phase of the cell cycle from S to G2 or from G2 to N. Okay, so this is how the regulations of cell basically occurs. So as I told, CDK and cyclins are regulated by certain proteins. Okay, and certain phosphorylation present in the CDK molecule induces the activation of CDK. Okay, so phosphorylation, for example, CDK induces mitosis by phosphorylating target proteins on serine and threonine. Okay, so serine and threonine basically are phosphorylated, and there can be inhibitory phosphorylation. Okay, which says that CDK should remain inactive, and there are some activating phosphorylation that which triggers that the CDK should remain active and can translocate into the next phase of the cell cycle. So here you can see that how the CDK is basically regulated. So cyclin basically helps in the activation of CDK. Okay, so cyclin helps in the activation. So if cyclin is bounded with the CDK, that triggers that the CDK will get activated. So overall, initially there are certain phosphorylation sites on the CDK, that is threonine 14 and tyrosine 15. Okay, so if this threonine and tyrosine are phosphorylated, then it triggers that the cells are CDK will remain inactive. But if there are certain phosphatase, okay, for example, as we talked V1, so V1 is a phosphatase over here, okay. So V1 is a phosphatase which will remove the tyrosine 14 and 15 phosphate, and it will help in the promotion of the mitosis, okay. So this is the regulation which is happening. So phosphatase as well as phosphorylation enzymes are also required, okay. So there are certain activating phosphorylation also, which is threonine 161. So if this residue is phosphorylated, then the CDK will remain active. Okay, and if these residues are phosphorylated, then it will trigger the activation. So certain cyclin-dependent kinase inhibitors bind to the CDK, okay, and induce the phosphorylation. So as I told you, inhibitory phosphorylation are done by this cyclin-dependent kinase inhibitor, okay, which induces the phosphorylation of threonine 14 and 15, and it negatively regulates the CDK. Okay, so is it clear to everyone? So now we can move to the next phase of the regulation in the
सर जो हमने फर्स्ट वीक में
yes.
क्वेश्चन सॉल्व किए थे वो
Uh,
असाइनमेंट क्वेश्चन से दे वर नॉट मैचिंग.
yeah, so actually that was different. That was some model questions. Okay, so that was a question from the previous year I took from the assignment.
ओके.
So currently, yeah, I also got aware in this week only that there are different questions. Basically, the questions get repeated, but in
ओके.
this session, basically the assignment questions are new. So we can also discuss that question if you wish. Okay, have you attempted that
Yes
assignment
sir.
question?
Yes sir, I have attended. That's why I'm telling that. That's why I had a doubt in one two questions, so that's why I was saying that please just can you just discuss them
Yeah,
also.
we can discuss that at the after this. Okay, so
Okay
we
sir.
can take that. We can take that also. Yeah, so as I told, there are CDC. So this is an example of CDC2. Okay, so here you can see tyrosine 15 and threonine 14 as well as threonine 161. So these two are inhibitory phosphorylation and this is activating. So V1 by cyclin activating kinase. Okay, so V1 will phosphoryl what V1 cyclin activating kinase with phosphorylate. The phosphorylate this threonine 161 and V1 will phosphorylate the Y15, okay, Y15 and 14 basically of the CDC2. So that is an inhibitory phosphorylation. So it will inhibit it, cyclin CDC complex will cyclin and CDC2 complex will remain inactive in the G2 phase. Okay, even though the threonine 161 is phosphorylated, but the inhibitory phosphorylation is there, then that cyclin CDC complex will remain inactive. But when you move to the CDC25, this is CDC25 is a phosphatase which removes the Y15, okay, which removes the phosphorylation from 15 and 14 and keeps the threonine 161 active. Okay, so now this cyclin CDC complex is active. Okay, and then if it is active, then that triggers that cells can enter from the G2 phase into the mitosis stage. Okay, M phase. So CDC22 regulation with CDC25 regulation uh, detects that whether the G2 cyclin will enter into the mitosis stage. Okay, and after that, when its role is completed, when the replication and the cell division has occurred, again the cell needs to undergo into the G1 phase. So this transition occurs by degradation of cyclin. Okay, so if the cyclin will remain active, uh, uh, then the cell will undergo premature mitosis. Okay, it will not undergo the G1 phase. So cyclin will get degraded and the phosphorylation, activating phosphorylation will get removed from the CDC2. Okay, and that way it will get triggered that the cell will again enter into the G1 phase. Okay, and when it enters into G2 and then the phosphorylation and all happens. Okay, during if he has to undergo into the mitosis state. So this is the one example of how the cyclin and CDK complex operate in the cell cycle regulation. So here you can see this is the CDK2. Okay, this is a structure of CDK2 and this is the cyclinic structure. So when cyclin is bounded with the CDK, there you can see that this portion, okay, this or yellow color portion and the blue color portion in this. Okay, so this yellow color and the blue color portion, the conformation is slightly changed, and due to this change in the conformation, it will expose the activating site on the CDK. Okay, that is cytosine 161, as well as the inhibitory site as well. Okay, so phosphatase and activating phosphorylation activating enzyme will work on this complex, and it will help in the limiting the it will help in the activation of the CDK2 cyclin complex. Okay, so this is the structural aspect of how binding of cycling helps in the activation of the CDK. Basically, it change the conformation of the certain activating regions of the in the CDK2 complex, CDK2 protein. Okay. So here you can see the examples of certain CDK and cyclin complex which are observed in the various states. So here the cyclic CDK complex, and here you can see G1 CDK, G1S CDK. So transition from G1 to S. Which are the CDKs involved? S CDK and M CDK, which CDK which are observed in the mitosis. So these are the vertebrate CDK cyclin complex. So in the G1 phase, basically CDK4 and 6 are present, and these CDKs are bounded with the cyclin D. Okay, and cyclin D, there are three cyclin D isoforms of cyclin D present: D1, D2, and D3 in the mammals. Okay, in the vertebrates. In the G1S phase, CDK2 is present, which is bounded with the cyclin E. Okay, the 2E complex will be get observed, and then if this complex is formed, then only the cells will transfer from the G1 to S phase. Okay. In the S phase, basically the cyclin 2, CDK2 cyclin A complex will be get formed, and in the AM phase, CDK1 cyclin B complex will be there. Okay. So this is the thing that the CDK1, if you mutate the CDK1, for example, okay, if you want to study that whether CDK1 is involved over in which cycle, you mutate the CDK1 and you observe the cell cycle. Okay, which cell cycle is getting hampered. So there you can observe that mitosis phase is getting hampered. So there you can see that then you can say that CDK1 is involved in the mitosis stage of the cell cycle. Okay. So CDK1 basically, which are observed in the vertebrates, that CDK1 is also known as CDC28 in Saccharomyces cerevisiae and CDC2 in S. pombe. Okay. So basically, in the yeast, whether it is a budding yeast or a fusion yeast, the CDK is only present in one. Okay, only there is only one CDK present in the whole cycle. Okay, from G1 to SM phase, only one CDK is present. That is in S series is CDC28 and in S form CDC2. Okay, it is not the case in the vertebrate where multiple CDKs are involved in the different cell cycle. But the cyclins are present different. Okay, so over here there are different cyclins present, but the CDK remain constant in the yeast. Okay, so that is one thing you need to remember in this case, and you need to remember that which cyclins are present in the in which stage. Okay, so that is also an important information from this slide. So overall, what we discuss is that there are for cell cycle transition from G1 to S, there are certain cyclin CDK complex which is activated, and it is sensed by certain proteins that whether the DNA is replicated properly or not. And if the DNA is replicated properly, then only the cyclin CDK complex will get activated, and it will transfer it into the next phase. Okay, to G2 phase. And after that, the cyclin will get degraded of that phase. And during the mitosis as well, if the proteins and all the other components are present, which helps in the mitosis. So during the mitosis, for example, uh, uh, during the mitosis, spindle fiber formation is an important stage observed in the mitosis. Okay, and whether the chromosomes are segregated equally or whether the spindle fiber is decell-lined. So if the spindle fiber are decell-lined, then also the cyclin CD it can also hamper the it can also limit the activation of the cyclin CDK complex. Okay, and it limits the degradation. It limits the replication stage, the division stage of the during the mitosis. And after the stage is complete, the cyclin gets degraded. Okay, and then it enters into the new phase of the cell cycle. Okay, so is that clear to everyone? This the case of cyclin CDK complex.
Yes, sir.
Yeah. Others? Can I get like
Yes,
other
sir.
others? Yeah. Yeah. So apart from cell cycles, okay, CDK cyclin complex, there are also checkpoints proteins present. Okay. So checkpoint proteins are basically helps in checkpoint proteins are basically the it activates the cyclin CDK or enables the cyclin CDK complex. Okay. They are called checkpoint proteins. The checkpoint controls are the signaling pathway that acts to delay the cell cycle when there are perturbations observed in the cell cycle events. Okay. So cyclin CDK complex, as I told you, there are certain phosphorylation and phosphatase and certain phosphorylation proteins which are present. Okay, and that proteins basically sends the environment, the condition of the cells, and then only can activate or inhibit the cyclin CDK complex. So it acts as a signaling relay that, for example, if the DNA is damaged or there is a spindle fiber, there is a misalignment or the spindle fiber is not attached properly to the chromatids, then the cell will sense that signal, okay, and the cell will send some signals by some certain proteins, and that protein will transfer the information to another protein, okay, which will activate the another mediator. It will activate the other mediator proteins, and that mediator will transmit the signal to the effector molecule. Okay. For example, if we talk about effector molecule, then cyclin CDK complex we talk about. Okay. So this is the transmission which occurs. Okay. It's not that cyclin CDK complex functional independently. These are the various other proteins are also involved, and these are called the checkpoint proteins. Okay. So checkpoints basically slows down the cell cycle. Okay. It allows the repair of the damage before proceeding. So these are important. And if you have a mutation in certain cases, okay, it will do the abnormal mitosis stage. So when we talk about the cancer, okay, in cancer basically there is an abnormal proliferation. So there are mutation in the checkpoint protein, okay, and because of this mutation, the cells will not sense the damage and it will continuously replicate, okay, even though without repairing the damage, and the DNA replication will not be regulated in that case. So because of the checkpoint, because of the inhibition, because of the mutation in the checkpoint proteins over there. Okay, so checkpoint protein plays an important role. And if the cell, there is also another condition that if the cells, first the checkpoint protein, what it does that it slows down the replication or the division and helps in the repair process. But if the repair is unrepairable, okay, if the damage is unrepairable, then the cell will go to apoptosis stage and it will undergo the apoptosis. Okay, so yeah, so these are the two main regulators of the cell cycle. CDK cyclin complex, which phosphorylates specific targets and drives the cell cycle forward, and checkpoint proteins, which act as a negative regulator, okay, which prevents the entry into the next phase until the preceding phase has been completed. So checkpoint proteins inhibits the CDK cyclin complex so that it does not remain activated, and it allows the cell to repair the damage. Okay, and if the damage is repaired, then the checkpoint, then cyclin protein complex get activated. It will uh, lead to the cell cycle division process, cell cycle division. Okay, yeah. So here you can see certain DNA damage checkpoints. How the DNA damage is sensed. Okay, so if there is a damage in the DNA, here you can see like certain proteins are getting activated, red 9, red 17, 24, MAC3P. Okay, so these are called checkpoint proteins. Okay, and these checkpoint proteins basically gets activated and it activates another set of proteins. Okay, red 53 and MAC1, which uh, which relays the information that there are relays the information to the cycling CDK complex that there are certain abnormalities present in the DNA molecule and that you did not need to get activated at that stage. Okay, so this regulation can occur at the start as well, during the DNA replication stage as well, and during the conversion from the metaphase to anaphase as well. Okay. So metaphase to anaphase transition is important cell cycle. In that stage, basically the sister chromatids are separated into two daughter cells. Okay. So that stage is highly regulated. So this three stage, this check if the DNA is damaged, then it holds this three stage and it leads to the repair of this stage. So checkpoint proteins are present in the G1, S and G2, all the three phase, as well as it is also present during the M phase. Okay, M phase as well. Okay, so metaphase to anaphase stage. So, for example, if I talk about DNA damage, uh, so if I talk about the DNA damage, then there are single-stranded DNA damage and there are double-stranded DNA breakage. Okay, so single-stranded are recognized by the ATR. This is occurring in the S phase. I'm talking. Okay, during the S phase, if the during the replication of the DNA, if there is an damage observed in the DNA, then during in the single-stranded DNA breakage, protein called ATR will bind, and if there is a double-stranded breakage, then ATM protein will bind. Okay, so ATR will activate the check one protein and phosphorylate it, whereas ATR will phosphorylate check one, whereas ATM will phosphorylate check two. Okay, and when these proteins are phosphorylated, it leads to the inhibition of CDC25 by phosphorylating it. So, do you know what is the role of CDC25? We already discussed in the previous slide. Anyone? What was the role of CDC25? You need to respond, okay? Otherwise,
Sir,
if I
elongation
will
of
talk on, yeah, go ahead.
elongation of the cell during.
Not elongation. Basically, CDC25 is a phosphatase. Okay, here we saw that CDC25. Okay, so this CDC25, what it does, it will dephosphorylate the inhibitory phosphate present on the Y15 and T14. So it will remove the inhibitory phosphate, and and it will make the CDC2 cyclin complex active. Okay, and that CDC2 complex will enter into the mitosis. Okay, so basically, what is happening over here is that there is a phosphorylation on CDC25, so it will not dephosphorylate the CDK2 and CDK1. Okay, CDK2 cyclin and as well as CDK1 cyclin complex. Okay, so CDK2 and 1 will not get dephosphorylated. So inhibitory phosphate will be present, and it will send then that way the cell will not be able to enter into the mitosis stage. Okay, so the cell will get arrested in the S phase only. And if the DNA is if the DNA damage is repaired, then this phosphorylation, then there is a dephosphorylation of this check 1 and 2, and there is also the dephosphorylation of the CDC25. And dephosphorylated CDC25 can dephosphorylate CDK2 and 1. Okay, and that can allow the transfer of the cell into the M phase. So this is how the regulation is occurring. Okay, so these are called checkpoint proteins. Okay, which acts on the cyclins, and cyclins are the master regulator over here, which regulates whether the cell should be entering into the next phase. So is this step clear to everyone?
Yes, sir.
Yeah. So next is P53 mediated mechanism. Okay, so P53 is activated. So P53 is an important protein. Okay, and if you had studied regarding the cancer, so P53 is a tumor suppressor gene. Okay, and mutation in that will lead to the formation of the tumor. Okay, so basically what it does is that P53, P53 can get activated by say this double-stranded break, okay, in the DNA. So double-stranded break will induce the activation of ATM, and it will lead to check-2 phosphorylation as discussed earlier, okay. And check-2 will lead to the phosphorylation of P53, okay. And P53, what it does, P53 is a transcription factor basically, okay. And P53, when it is phosphorylated, it binds to the DNA sequence and it transcribes the P21 mRNA. So P21 mRNA, P21 mRNA codes for a protein, P21 protein, and P21 protein inhibits the cyclin 2 CDK2 cyclin E complex. Okay, so in the mammals, which are the CDK cyclin complex which are present in the G1 S phase as well as the S phase. Okay, so CDK2 cyclin E complex is present in the G1 phase, G1 and S phase. Okay, and if this P21 basically inhibits this CDK cyclin complex, so that is how the cells will not move from the S phase. Okay, so cells will get arrested in the S phase only, and it will not move to the G2 phase. Only if the DNA is repaired, then only it will move to the G2 phase. Okay, but if there is no DNA damage, okay, and the DNA is active, okay, if the DNA is intact and there is a proper replication occurring in the cell, then the check2 will not be phosphorylated and it will remain as a dephosphorylated, and that way the P53 will not be getting phosphorylated. Okay, so unphosphorylated P53 then undergoes the ubiquitin mechanism. So ubiquitin will be tagged onto the P53 protein, and through the proteasome system, the P53 protein will then degraded. Okay, and because the P53s got degraded over here, uh, the P21 will not get transcribed, and the cell can undergo the transformation from the S phase into the G2 and M phase. Okay, so if the levels of P53 are high, then you can say that there is a DNA damage occurring, and the cell cycle arrest will be present. But if the levels of P53 are low, then there will be continuous synthesis of that the cell will undergo the division process. Okay, in the mitosis stage, it will undergo the mitosis phase, and it will undergo the G2 and mitosis stage. So this regulations is important in governing the cell cycle. Okay, so in cancer cell, basically what happens is that uh, uh, the P53 does not sense the DNA damage. Okay, so even though the DNA damage is there, P53 does not get phosphorylated. Okay, so it is called tumor suppressors in gene. So basically there are two sets of gene. One is called oncogene and one is a tumor suppressor gene in the cancer. So oncogene, what it does, oncogene are classified as a gene, as a protein, uh, the activation of which leads to the promotion of cell cycle. Okay. So for example, when I talk about the simple example, for example, activating phosphorylation on CDK cyclin complex. Okay. For example, threonine 161, that is an activating phosphorylation. Okay. So if the phosphatase does not remove the phosphate group from there, then it will remain constitutively active. And if it remains constitutively active, then the cell will undergo multiple phase of division. Okay, without any regulation. So that is called oncogene. Okay, the activation of which remains constitutive and it leads to the proliferation of cell. Reverse to that. Okay, in contrast to that, tumor suppressor genes are the genes whose inactivation uh, leads to the promotion of the cancer. Okay, promotion of the cell cycle and cell division. So here you can see that if the P53 is degraded, then there will be no cell cycle arrest occurring. Okay, so P53 in ideal scenario, P53 should be degraded. Okay, then only if the DNA damage is not there, then P53 will get degraded. But if the DNA damage is there, but then also the P53 is getting degraded, at that stage it is toxic to the cell. Okay, it is the regulation is hampered in that case, and at that stage there will be uncontrolled proliferation. Okay, without any checkpoint control in the cell. So that is how the P53 is a major tumor suppressor gene which are present uh, which are found to be inactive in case of certain kinds of tumors. Okay, so this is how the mechanism occurs over there. Yeah, so basically, what we can do, they after this one slide, we can see some assignment question, and later we can discuss the ubiquitin mediated proteasome degradation. Okay, how it is happening. So this is one case study. I mean, one experiment which was done, and whether to check that DNA replication checkpoint. Okay, how it is functioning in the S phase. So they wanted to check that in the S phase, if the DNA is not getting regulated, if there are the checkpoint proteins are irregulated, then what is the observed effect observed? Okay. So first thing is they use the untreated cell in which the normal S phase, G2 and M phase are present. And over here you can see that the DNA was replicated properly. There was proper S phase, G2 as well as M phase, and the two daughter cells are present. Then they use the caffeine. Okay, so caffeine is basically uh, molecule, okay, which uh, which helps in the transformation from the G2 to M phase, okay, which irregulate the G2 to M phase basically. So as I told that certain CDK cycline complex should remain active, but caffeine what it does, it bypass the regulation present, okay. So if the CDK cycline complex will not be present and there are other abnormalities present, then also it will enter into the M phase, okay. It will not restrict into the G2 phase. So there you can see that even though there also you can see the normal replication of the daughter strand because we have not inhibited the synthesis phase. Okay, the synthesis phase is active. Only we targeted the G2M phase by caffeine. So it targets the G2M phase, and here we can see that the DNA is replicated properly. But if we target the ACE phase, okay, so by the hydroxyurea, okay, 5-hydroxyurea. So hydroxyurea, what it does, it inhibits the replication fork movement. Okay, movement of the replication fork, so that the DNA synthesis will not occur. Replication of the DNA will not occur. Okay, so hydroxyurea inhibits the DNA synthesis, and basically, what it does is that if they treated with the hydroxyurea, there they observed that the cell did not undergo the mitosis. Okay, so here you can see that because the DNA was not getting replicated, because of that, the checkpoint protein got activated, and it responded that the cell should not undergo the mitosis in this case. Okay, so here you can see that what is the importance of cell cycle regulation in the S phase. Okay, and now in the fourth condition, what they did, they used the hydroxyurea plus caffeine. Okay, so as I told that caffeine caused the deregulation of the G2M phase. Okay, so even though the DNA will not get replicated in the S phase, the cell will undergo the mitosis. Okay, because there will not be any regulation during the G2 and M phase cell cycle. Okay, and the cell will undergo mitosis. It bypasses the SG2 as well as the G2M phase of the cell cycle. So mitosis will be present. So as the cell DNA is not getting replicated, okay, even though the dot mitosis is there, that is called suicidal mitosis. Okay, and during this mitosis stage, the DNA unequal composition of DNA replicate DNA will be present in the daughter cell. Okay, so from this experiment, you can say that what is the importance of the checkpoint proteins present in the S phase. Okay, so if you inhibit the S phase, there is a check, there is a delay. There is if you use the certain inhibitor which hampers in the replication, then checkpoint protein will get activated, and it will not lead to the mitosis stage. Okay, but if you use certain reagents, okay, or certain molecules which bypasses this checkpoint protein, okay, which limits this checkpoint protein, then you will get the aberrant mitosis present. So this shows the importance of the checkpoint proteins present in the S phase. Okay. So cell cycle regulation. So is it clear to everyone? This Steve? Is there any
Yes,
doubt?
sir.
Yeah. So, so overall, if I talk, then there are cell cycle checkpoints. Basically, it helps in the regulation during the certain stages. During the G1 to S transition, if there is a DNA damage, okay, then it will limit the progression. In the S phase as well, it will limit the progression to the G2 phase if there is a DNA damage, as well as in the G2 to M phase as well, it will stop. And in the mitosis phase as well, during the spindle assembly, during the mitosis to anaphase transition, there also it will get the spindle cell cycle checkpoints will work over there as well. Okay. This is the overview of that thing. So now we will solve some more questions, okay, for assignment. So then we can talk about if there will be time, then we can talk about the ubiquitin process, okay, ubiquitin system. So what should I do? I mean, if you have not basically answered the assignment, okay, you have not gone through the assignment of the second week. So what should we do? Should we discuss these questions over here? Or
Yes, sir.
yeah, so we can discuss it, okay, and after that you can go through the assignment. You can solve over here, okay. You can try. I will not reveal the answer. Depending on the like information what I taught to you. Okay, in this lecture, you can like answer the question. So one thing what we can do on this assignment, because this assignment should be solved by your own, okay, and there is a grading observe in the assignment tab. There is also a grading, okay. So if you solve over here, then if you apply over there, then it will be problem. So one thing, as I told it previously, that if you go through the lectures and solve the assignment before this class. Another thing is that I can come up with the questions apart from this original assignment question. It's a model question, okay? So that, but in that case, basically, we will not be able to discuss the assignment question in the next class. So that is the issue, okay? Yeah, so answer this question. Which of the following is correct regarding the mitosis promoting factor? So first is induces the oocytes to become an egg. The biochemical verification reveals MPFF2 subunits and CDK is a regulatory subunit which activates cyclin. Okay. So mitosis promoting factor is a CDK cyclin complex. Okay. So which of the options is correct from these? You need to answer. Yeah, please go ahead. Please respond. Okay. So basically, what should you do? You can take pen and a paper and you answer it. Okay. Honestly, you can answer and you can grade yourself. Okay. And if you are making any error, then you can we can explain it. Okay, after solving the question, I can also explain the question. So in assignment also you can just over there also you can go through the question like this only. So here you can solve and you can grade your own. Okay, depending on that. So what will be the correct answer for this?
Sir, 1 and 2 are correct, sir.
One and two. Option A.
Yes, sir.
Others. Please respond. Please give at least two, three answers. Okay, so that 3 to 4 answers from different individuals, so that then only it will be interesting to solve this question. Otherwise, I will just reveal the answer. Then it is of no use. Okay, you should also at least try. Okay, and so that it could be fruitful discussion from both the sides. Any idea? I mean, any answers from others?
Sir, they're responding in the chat, sir.
Okay, in the chat. So you can please on your microphone and you can answer. Okay, in the chat I will not be able to see. That is the issue because I'm using only one screen. Okay, so you can on your microphone and talk. Okay, I'm not telling you to on your video. You
Sir,
can.
option A, 1 and 2 is correct.
Hmm. Yeah, so option A is correct. Basically, the mitosis promoting factors are the CDK cycline complex. Okay, and it induces the oocyte into the M phase. Okay, and it has a two subunit that is CDK and cyclin. And CDK is a regulatory subunit which activates cyclin. No, it is incorrect. Cyclin is a regulatory subunit which activates the CDK. Okay, so option 1 and 2 is correct over here. Second question is upon activation of CDK1, which of the following events are likely to occur?
Sir, it'll lead to mitosis, dissociation of chromosomes from spindle fibers.
Dissociation of chromosomes from the spindle fiber. Okay, others? So if you're answering in chat, then I will not be able to see. So please on your microphone. So actually over here the answer is option C. Okay, it leads to the dissociation of lamine dimers. So CDK1 basically what it does, CDK1 there is a during the G2 phase of the cell cycle. Okay, during the transition of the G2 to M phase during the alignment phase. So it degrades the lamine. Okay, so lamine are the proteins which are present in the nuclear membrane. So you need to disrupt the nucleus, then only your chromatids will get exposed into the cytoplasm. So CDK1 it dissociates the lamine dimer. Okay, and for the dissociation of chromosome. Okay, so dissociation of chromosome basically happens by another protein. Okay, which is called separase. Okay, separase is a protein which leads to the dissociation of chromosome from the spindle fiber. Okay, so that will be discussing further how it happens during the mitotic mitotic assembly complex. There is a protein complex, okay, which activates the separase and activates the dissociation of chromosome from the spindle fiber. Okay, but the lamine dimers are dissociated by there, and formation of nuclear envelope. So that is incorrect. It basically leads to dissociation, okay, of the nuclear envelope, and it leads to the releasing of the chromatids into the cytoplasm. So option C over here is correct. So a third question, suicidal mitosis is most likely to occur in the which stage?
Sir, in presence of HU and caffeine.
Okay, others? This is rather
Will
simple
be
question for you, sir. Yeah,
last option, sir.
yeah, option 4. So option D is correct. The presence of HU and caffeine. So that we already discussed. Okay, so basically caffeine does not allow the regulation, and HU limits the replication. Okay, so that will if you use both the system in a single system, both the reagents, then it will lead to suicidal mitosis. So replication will not DNA will not be segregated properly. So option 4 is in S Pombi, the homologue of CDC28 is known as.
CDC2.
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 Yes, sir. CDC2.
Yeah. So option B is correct over here, CDC2. So this table you can see. So CDC2 basically, okay, CDC2 which is present in the S. pombe, and CDC28 is present in the Saccharomyces cerevisiae. Okay. In S. pombe, CDC2 will be present over here. Is there any question? Yeah, someone has raised hand. Yeah, any doubt? Vani Shri. Is there any doubt? Person who raised the hand. Okay. Yeah. So, fifth question is, what is likely outcome when cytoplasm from the interface cells is injected into the oocyte?
Sir, option A.
Sir, option C.
Option C and option A. Others?
Option A, sir.
Okay. Any other response? Okay, so option A is correct over here. Oocyte remains in the G2 phase. So the option C would be correct when the cytoplasm from the M phase cells will be injected into the oocyte. Okay, then only the oocyte will be transferred into the M phase. If the cytoplasm from the interface cells will be transferred into the oocyte, then it will remain in the G2 phase. Okay, it will not transfer into the M phase because interface cell will not contain the proteins necessary for the transfer of the oocyte into the M phase. Okay, only when the might MPF would be present, that is mitosis promoting factor will be present, then only the it will promote into the M phase. So you need to be clear in both the cases. Okay. The sixth question is, so if you have not attended the lecture, then it will be difficult for this to answer. But like if you know earlier, right, if someone have attended the classes of the week, okay, all four classes, then they can give an answer. So here the following part of the yeast proteasome system unfolds the protein to enter inside the proteasome.
Sir, option B.
Option B. Okay. Anyone others who have not attended? I mean, who have attended and can solve, or if you have prior knowledge, then also you can answer. So option B is correct. That is a AAA ATPase subunit. Okay. So basically, this is a proteasome system. Okay. So in yeast proteasome system. So yeast proteasome basically contains of three subunits. Okay. There are three subunits present over here. So the middle portion, which is called a core particle. Okay. So in middle portion, it is a 20S proteasome system. Okay. And in that, your protein will get degraded. Okay. And your ubiquitin will get separated from the tag and your protein will get degraded. Okay. Whereas in this pink color module where you can see it is a triple A ATPase module. Okay. So what it does is that first let's talk about the regulatory subunit. So regulatory subunit will recognize the ubiquitinated protein. Okay. So protein which is ubiquitinated at the end terminal, it will get recognized by the this regulatory subunit, and it will translocate it inside this regulatory subunit. Okay. And when it translocate inside, there is this module known as AAA ATPase module. So this module basically what it does, it leads to the formation and deformation of ATP molecule. Okay. So why it is doing to generate the ATP to generate the energy. Okay. So first they generate the ATP molecule, then it will degrade that ATP molecule. Okay. To generate the energy to push that protein inside the core particle. Okay. The energy derived from that will allow the protein to enter inside the core particle where it is getting degraded. Okay. So beneath this triple ATPase, here you can see the yellow color portion, which is a part of 20S proteasome system. Okay. So this yellow colored portion, a domain that removes the ubiquitin-ylated tag from the protein, and the 20 and core particle will degrade the protein. Okay, which is ubiquitin-ylated in that system. So this is the whole proteasome system will work, which works over there in this case. Okay, and it is present as a dimeric form over here. Okay, so there will be two subunits of regulatory subunits and two triple ATPase subunits. Okay, and in between here you can say 20S proteasome system, and the regulatory subunits are also called 19S proteasome. Okay, this is 19S and this is a 20S in the middle. So is this clear? So basically, if I talk about core particle helps in the degradation of protein. Okay, and for triple ATPase will lead to the unfolding the protein. So energy which is provided by this ATP generation and hydrolysis, it will also push the protein to the proteasome, the core particle, as well as it will also unfold the protein. Okay, from this tertiary structure. So that it will lead to the formation of primary structure, and then it can be degraded to the core particle. Okay, so this triple ATPase also leads to the unfolding of the protein and leads to the entering into the proteasome system. So this is clear.
Yes, sir.
Yes, sir.
Yeah. So this you can solve this question. Number 7. Which of the following is correct about the cell cycle? There are 4 statements, and you need to pick one statement which is correct. So you can take your own time. Okay. We can wait 2-3 minutes, and you can go through each and every sentence clearly, and then you can give the answer. Yeah. Anyone? Have you solved? We need more time, or you are not getting the question?
Sir, I think it is option D.
Option
Sir,
D.
it's B or D. I'm not sure. It's B or D.
Okay, so B is in mammalian cell, the R or the restriction point in the G1 is analogous to start. And option D is CDC28 is the yeast cycline that plays a critical role in the regulation of the cell cycle. CDC28 yeast cycline. So over here, actually, you need to look very properly into the each and every term. Okay, so if there is a slight change in the term as well, you even if you will not observe the thing, okay, you will say that yeah, this may be correct, but a single word, uh, if you don't pick that single word, then your answer will get incorrect. Okay, even though you not knew that thing, that this would be I knew about that thing. Okay, so we will discuss that. So what about others? They have solved. Uh, Can you respond? Can I get like two, three response from others?
So can you please explain this? Means all the four terms, four options.
Yeah. Okay. Yes. So first one is in S cerevisiae, after reaching to a certain size, not reacting. There is a writing error over here. Reaching to a certain size, at the start, the cells gets committed to entering the G2 phase and ultimately completing the cell cycle. Okay. So in S cerevisiae, basically, it's not observed. In S pombi, it is observed. Okay. In S cerevisiae, basically, the budding, it occurs through budding. Okay. The replication occurs through budding. In S pombi, basically, it reaches to the certain size and then it enters to the G2 phase and it completes the mitosis phase. Okay. So this is incorrect. Second is in mammalian cell, the R or the restriction point in G1 is analogous to start. Yes, the start which we called in the yeast. Okay, so start phase is also called the restriction point. Okay, in the mammalian cell. Uh, so this option is correct. The G1 phase, okay, start phase is also called the restriction point in the mammals. The third option is temperature sensitive. CDC28 can bud at the non-permissive temperature. No, basically CDC28 temperature sensitive CD28 will not allow the replication of the DNA. Okay, it will hamper the DNA replication in the S phase only. Okay, so that mutant will not be able to bud. Okay, because there will be mutation in that DNA synthesis will not occur. Okay, and CDC28 is a yeast cycline that plays a role in the regulation cycle. CDC28 is not a cycline; it is a CDK. Okay. CDC28 is a CDK, cyclin are others. Okay, cyclin are others. There are cyclins such as CLN, okay, in the yeast. So in this table, you can see that table which I had shown earlier. Okay, so this one. So CDC28 is a cyclin, CDK. And cyclins are this one, CLN3, CLN1, 2, 5, 6, and 1, 2, 3, 4, CLB. Okay, so that you need to remember carefully. I mean, look at the terminologies as I told. What is CDK? Which are CDKs? Which are cyclid? You need to remember very accurately this thing. Otherwise, you can have incorrect question. Okay, so in G1, basically, here you can say restriction point present. Yeah, the question 8. This is a simple whether which of the following may complement the defect in the CDC2 mutant.
Sir, human CDK1.
Okay, option B. Others, So option B is correct. CDC2 is a complement of human. Okay, human cDNA complement. Uh, so CDK1, basically what they did was they took the CDC2 gene, okay, and they took the human cDNA library, and they wanted to check that whether any complementation can occur in the by the cDNA library. Okay, so they found that the CDK1 gene hybridized with the CDC2 gene. Okay, so that from there they found that CDC2 is a complement which are present in the CDK1. So basically the idea is if you inject the CDK1 inside the CDC2 mutant, okay, so the CDC2 mutant will undergo the mitosis stage. Okay, so as I told, CDC2 codes for P34 protein, and P34 leads to the mitosis in the S form B. Okay, so the CDK1 will complement the CDC2 mutant and it will lead to the mitosis of this S form B cell. Yeah, so this is this question: addition of hydroxyurea to cell results in uh, which uh, which of this thing? So in hydroxyurea, what I told was that it inhibits the replication. Okay, so basically it inhibits the replication by uh, decreasing the concentration of deoxynucleotidal phosphate. Okay, DNTPs. So it decreases the DNTPs by targeting a certain enzyme called a ribonucleotide reductase. Okay, so ribonucleotide reductase is an enzyme which converts the ribonucleotidal phosphate RNTPs into the DNTPs. Okay, and if that enzyme is phosphorylated, if that enzyme is inhibited by hydroxyurea, then DNTPs will not be formed and DNA replication will not occur. Okay, so hydroxyurea acts by that mechanism. So in this case, like first option is incorrect. You will not. First option is will not. The answer is not the first. Okay, increase DNTP. It will basically decrease the DNTPs. So out of three, now you have to choose which is the correct.
So there will be a decrease in RNTPs.
Okay,
So B,
others.
B, degrees R&D, please.
Others. Can I get two, three response? Okay, so correct answer is none of the above. Okay, so it will not decrease the RNTP. Basically, hydroxyurea work on the ribonucleotidal reductase. Okay, so ribonucleotide reductase, as I told, converts the ribonucleotide into deoxyribonucleotide. It will not decrease the RNTP. RNTPs will remain constant, but it will decrease the DNTPs. Okay, by limiting this enzyme. So increased, if there would be increased RNTP, then that option will be correct. But decreased RNTPs will not be you will not observe the decreased RNTPs. Okay, over here. So option E is correct. None of the above. Last question: when shifted to the restrictive temperature, CDC28 mutant cells. So this is rather simple, which you already discussed it.
Sir A.
Hmm. Others? Okay, so option A is correct. Basically, CDC28 mutant doesn't initiate the DNA synthesis. Okay, as a result, the cell get arrested without the budding formation. Okay, CDC28 is analogous to CDK1. Okay, in the Acromasis cerevisiae. So, so yeah, that's all from this assignment question. These were the assignment questions, and like rather you have solved over here. And although due to like time constraint, we have 2 hours. Okay, so in 2 hours we cannot. I cannot discuss the whole week content. I can just brief you about the content. What are the main important points? Okay, but as I told in the starting of the lecture as well, okay, if you attend that classes before this class, okay, if you attend that content, week content, then you come to this class. So then, for one thing is that you will get your complete revision of also from this class, okay, of that week. Another thing is that you will also be able to solve the assignment question, and the assignment question solving will be rather interesting from your side as well. Okay, so that is the important that if you just watch one lecture per day, okay, so there are one lecture maximum it is like 30 to 35 minutes, okay. Some lecture might be having 50 to 50 minutes, but most of the lectures are like 30-35 minutes only. So in a day, if you like at the end of the day or in the middle, if you just watch one lecture, then also you can complete that lecture in the 4 days, okay. And on the fifth day, for example,
So,
Monday,
can you
yeah,
discuss first week's assignment? The
yes,
assignment
yeah,
questions of the first week because the questions weren't matching.
yeah. So mm, that that I have to open because just a minute. So these are the questions for week one. So which questions you had?
Or flip flop question.
This one. Which of the following phospholipid molecule movements the plasma membrane is rare? Okay, so
Yeah.
lateral diffusion, rotation, flip flop, and flexion. So flip flop would be rare, no? That I already discussed in the class as well. Flexion means the movement from the plasma membrane, the lipid molecules. Okay, so these three would be present: lateral diffusion, rotation, and flexion. Flip flop movement will be rare. That is only observed in the apoptotic condition. Okay.
Okay.
Is it clear to everyone? Like, is there any doubt, other doubts you can ask?
Second one, sir.
Sorry,
Fourth
third,
question, sir.
third, sir. Which one are following characteristics of prokaryotic cells?
Prokaryotic cell,
Nucleoid.
it is, so what will be the answer? It is not multicellular. Presence of
Maybe
mitochondria, no.
presence of
Mitochondria is,
nucleoid.
Nucleoid, sir.
yes, presence of nucleoids. So this is the correct option. Okay.
And fourth one, sir.
Fourth question, sir.
Which of the following can be considered as having a polar side chain? So polar side chain, which of the protein? Alanine, valine will not there. Lysine, histidine,
Serine,
serine, aspartate,
Serine and tyrosine, sir.
serine, tyrosine. Okay. But what about this aspartate and glutamic? Aspartate and glutamic acid, I think, would be the
Sir, asparagine and glutamine, I think they have polar side chains.
acidic side chain.
What about the
Okay,
first one,
so
sir?
Sir, I think
first
it's
one,
serine and tyrosine.
serine inside, serine, tyrosine. Okay. Yeah, but I have doubt in this aspartic and glutamic because that serine and tyrosine is there, but that is polar uncharged. Okay, serine
Okay,
and
sir.
tyrosine are polar. There are two kinds of polarity. Okay, polar uncharged and polar charged. So lysine histidine is a positively charged. Aspartic and glutamic acids are like negative charge. Okay,
Okay, sir. Sir,
so
but
they
in
are
the
also
lecture it was mentioned as uncharged only, sir.
so if actually the question is not framed properly over here. So they need
Yes, sir.
to show that polar uncharged side chain. Then you can consider serine tyrosine.
Okay, sir.
Polar uncharged, but polar charge, then there we can have two options. So basically these three, all the threes are polar side chain, apart from alanine and valine. Okay.
Okay,
So
sir.
I mean there
What
is
Thank
is
you,
the
sir.
first one, sir?
here the following holds true for the cell. Cell era is just spontaneous generation. Cell carry and pass on hereditary information. So this would be the correct.
Second option.
Cells are dynamically closed system. All of the above.
Sir, it's B na in first.
Yeah, but what about this option? Cells are dynamically closed. So
Sir, all of the above तो नहीं होगा ना.
all of the above, I mean, the A option, this option, I don't think it is correct. Cells arise as spontaneous generation.
Spontaneous generation.
Spontaneous
Yeah.
generation, I mean, cells will only replicate from other cells. Okay, so it will not
Yes,
be
sir.
spontaneous generation. And this, I am little confused on this. Cells are a dynamically closed system.
Closed system.
So basically, the cell cycle regulation occur, but there might be other factors as well. Okay, that might be so that might be regulated, okay, from the outer cells, outer environment. There are other certain factors as well, which are involved in the replication and basically uptake process. So certain, for example, if I talk about glucose or other soluble factors, okay, so that are soluble factors that are taken from the outside and it is translocated into the cell by transporters and various other plasma membrane proteins. So that cells are closed. We cannot tell that cells are closed system because it is an open system. It can interact with the outer environment as well. Okay, so this cannot be a correct option. So in that case, this is the correct option. Cell carry and pass information to the daughter cell. Option B. Is it clear to everyone?
Yes, sir.
Sir, I have a doubt in that flip flop one. I didn't got your point because flip flop is the phospholipid um, is the most common. Like I think flip flop, how it can be the
No,
rarest.
flip flip flop is not common. Flip flop is rare. Okay, so if I can open the slide for you, I mean, in the previous lecture also I had discussed that thing. Okay, but we can look. Flip-flop movement will only occur when there is a certain conditions, okay, abnormal condition. So here you can see flexion is the movement of this acyl side chain, fatty acyl side chain. Okay. Lateral diffusion means the plasma membrane which get laterally diffused. Rotation means the head group, polar head group, which rotates on its own axis. And flip-flop is rare, which occurs between the
Okay, sir.
two inside and outside side chain. So this is clearly mentioned over here.
Okay, sir. Thank you, sir.
Yeah. So yeah, it's if you don't have any doubt, then we can like end this session, and from the next session, like I will recommend that. Yeah, is there any question like Vanishri? Someone is raised, and I don't know when it was raised, but if you don't have a question, then yeah. So thank you everyone for attending this class. And as I told ki, in the first lecture as well, if you go through the lecture first and solve the assignment on your own, then this session will be fruitful to you. Okay, so if you can spend half an hour for this class every day, okay, when you go to sleep or in between your classes, half an hour if you keep aside for this course, then in a every day, okay, on consistent basis for 5 days, okay, Monday to Friday. Then on Saturday we can discuss the doubts and clear the things. Okay, so that way
Thank you,
it will
sir.
be, yeah, good. So, yeah, thank you everyone.
Thank you, sir.
Thank you, sir.